Our second speaker in this session is Xiao-Jing Wang, who did her MD and PhD training in China and then proceeded to get additional training and to run laboratories in Texas, Oregon, before she moved to Colorado, where she currently is doing her work. And if she were here, I'd invite her onto the stage.
First, I'd like to thank the organizers to invite me to come to this great meeting. What I'm going to show you is the study related to head and neck cancer, but I have to warn you first, different from the speakers that show you lots of beautiful pictures, and I have lots of ugly pictures here. So head and neck cancer, as shown here, is the sixth most common cancer in Western countries. In developed countries, and the incidence is even higher. The 5-year survival rate has not been improved in the past 20 years. And from this picture, you can imagine that the primary tumor is a major problem to take care. And related to the poor prognosis, there's a significant shortage of in vivo models that mimicking head and neck cancer. So it's hard to test the novel approach for therapeutics when all other cancer types now move to the experimental therapies, and head and neck cancer is still at the stage of the making model. The technical difficulties to make genetically engineered mouse models for head and neck cancer is that head and neck epithelium do not have their own tissue-specific promoter. So whichever promoter targets to head and neck epithelium also targets to skin epidermis. But in most cases, the promoter activities are so much stronger in skin epidermis, and then the mice ends up develop skin tumor long before they develop head and neck cancer. So to overcome this problem, we developed an inducible transgenic system, which is very similar to the TED-ON system, except that the inducer is a RU46. So in a bigenic system, when the transactivator is targeted to the keratin promoter, if we swap RU46 in oral cavity, then the skin will not be affected. Only head and neck epithelium get affected. The first transgene we tried is TGF-beta1. Not only this is our favorite molecule, and also when we look at the expression pattern of TGF-beta in head and neck cancer, we saw that TGF-beta is increased in cancer, but also in adjacent mucosa, so indicating that TGF-beta overexpression is a relatively early event in head and neck cancer. What does this mean? And this is the reminder of the TGF-beta signaling pathway. Eric already introduced you to this pathway, so I just wanted to remind you that mutations, most of them are the loss of functional mutations of TGF-beta receptors. SMAD2 and SMAD4 are common in human cancers, so indicating this is a very potent tumor suppressive pathway. But in contrast to the loss of the signaling components, the ligand is overexpressed. So at least at the early stage, and people suspect that this is a negative feedback trying to reinforce the growth inhibitory effect of TGF-beta in cancer. However, when we overexpress TGF-beta in head and neck epithelium, now you can see instead of predicted growth inhibition, we saw the major phenotype is this tremendous angiogenesis and inflammation. Then the epithelial layer is tremendously hyperplastic and hyperproliferative. So we suspect that this is a secondary effect of inflammation and angiogenesis that feeds lots of positive growth regulators back to the epithelium layer. And to prove this, we separated the epithelial layer from the stromal, then cultured keratinocytes. Now you can see that in cultured dish, then TGF-beta transgenic keratinocytes indeed undergoes growth arrest, but it's completely override in vivo. So this is one of the best examples. I recruit students to work on animal models, in vivo models, to fully understand the biology of the molecule. But nevertheless, these mice did not develop any spontaneous tumor, regardless the severe inflammation and angiogenesis microenvironment. So it indicates that genetic insults is needed. And to look for these genetic insults, we look at the two molecules. One is the TGF-beta type 2 receptor. TGF-beta type 2 receptor loss, deletion, occurred about 50% in head and neck cancer. But when we delete TGF-beta type 2 receptor in head and neck epithelium, this is use Cre-Log system, again use RU46 as an inducer. We didn't really see any tumor formation. This is not a complete surprise because TGF-beta receptor loss often occur at the malignant state, so it needs an initiation event. The most common initiation event in human cancer is KRAS mutation, and then this is a mouse line produced by Tyler Jack's lab. So unlike the lung cancer, if you induce KRAS mutation in the lung, then they can develop to lung adenocarcinomas. But in head and neck epithelium, if we activate the RAS mutation, they can develop benign papillomas but never progress to malignant tumor. Nevertheless, this suggests that RAS activation can be an initiation event. But when we couple these two together, now you can see the mice develop head and neck cancer with full penetrance. So this is from the buccal, the tongue, esophagus, and forestomach. This is another reason I just showed you the patient's photo earlier. Now you can appreciate at the pathological level how close the model mimic human cancer. And they're all squamous cell carcinomas. They can develop to metastasis, but most of the mice like this died of the primary cancer. So in addition to the molecular alterations found in human cancer, head and neck cancer, also found in this model. One interesting finding is that TGF-beta ligand is increased in this model, which related to TGF-beta receptor loss. So we think this, again, is a compensatory mechanism, and it's getting higher in squamous cell carcinomas. But in tumor epithelium, because the receptor is gone, so TGF-beta cannot signaling and then cannot induce CDK inhibitors, for example, P15 or P21. However, in the stromal, TGF-beta is a secreted protein, so once it's secreted to the stromal still have intact TGF-beta receptor and SMADs. All these inflammatory cytokines, chemokines that are normally induced by TGF-beta is still increased in the TGF-beta receptor knockout tumors. So this shows that TGF-beta receptor loss almost play double jeopardy. In tumor epithelium, it blocks TGF-beta induced tumor suppressive effect, but enhance the paracrine effect that promotes tumor microenvironment. So the next question we ask, will loss of signaling SMADs have the similar effect? And this is the SMAD expression patterns in squamous cell carcinoma. We look at the head and neck cancer and also skin cancer. It's pretty much the same trend, that SMAD2 and SMAD4 are commonly lost, whereas SMAD3 is largely retained in most of the tumors. So we delete SMAD2, use the same system. If we induce the SMAD2 deletion in oral cavity or in the skin, and then they didn't really develop any tumors. However, if again, I mean, because SMAD2 loss is about the same time as TGF-beta receptor loss, so it suggests this is not an early initiation event. But if we expose these mice to a chemical carcinogenesis protocol, then the mice develop more tumors and more malignant tumors. The striking phenotype is that these tumors are intend to undergo EMT, and they develop spindle cell carcinomas, which is not very common in wild-type mice. And when we look at the markers, the E-cat herring loss occurred quite early in benign tumors. This is a wild-type tumor, largely retained E-cat herring, but SMAD2 knockouts, they lost E-cat herring. And the repressor snail normally is expressed in the cytoplasmic compartment of the wild-type tumor, but now get activated in the nucleus. And twist is another suppressor of E-cat herring, normally is expressed in the proliferative cells of the benign tumor. And now you can see they are activated in the nucleus as well. So we also look at several other EMT markers. They're all elevated, but among them, snail is the early event caused by SMAD2 loss. In fact, it's so static. And sorry, yeah, thank you. Sorry. So in human cells, normal keratinocytes, if you knock down SMAD2, and it's sufficient to cause nuclear staining of snail, and then the keratinocytes lost E-cat herring and undergoes EMT. But at the same time, if you knock down snail, and then it can reverse the phenotype. So indicating snail is one of the major player to induce SMAD2 loss associated EMT. So initially, this is a very surprising finding, because from previous speakers' talk, you will know that TGF-beta is a very potent pathway to induce EMT. You would expect that loss of SMAD should abrogate that phenotype instead of enhanced EMT. But based on the data I showed you earlier, then you can see that naturally occurring that we will think maybe TGF-beta 1 is increased, then it can signaling through other SMADs to induce EMT. And, but we really didn't see any change of TGF-beta ligand level. And then the next, we look at if SMAD-independent TGF-beta signaling is increased in these cells, because, for example, the MAC kinase and Notch pathway also related to EMT. But we didn't really see a change between wild-type and SMAD2 knockout cells. And in the end, a grad student looked at if the snail promoter occupancy by SMAD3 and 4 are increased in SMAD2 knockout cells. So the answer is yes. So the snail promoter has two SMAD binding elements. And you can see when SMAD2 is lost, then of course the binding is lost. SMAD3 binding did not change that much, but SMAD4 binding is significantly increased. And this happens in another site as well. So, but the question is the higher binding really translate to a higher transactivation? The answer is yes. So again, this is the cultured keratinocytes, human keratinocytes. So if we knock down SMAD2, then snail level is increased. But if we knock down SMAD3 and 4 together with SMAD2, then you can see the effect get reversed. And based on these data, we suspect that normally TGF-beta induce the SMAD complex that binds to the snail promoter. SMAD2 can also recruit repressors, so it's a balance between transactivation and repression. But when SMAD2 is lost, then SMAD3 binding is not changed. However, there's no computation between SMAD2 and SMAD4. So there are increased SMAD4, and also there's no one recruits repressors, and this complex may be more potent than this complex for snail transactivation. And then this brings up another controversy that shows that SMAD4 activation is actually pro-oncogenic. And knowing that SMAD4 is a potent tumor suppressor, and this is a little bit puzzling. However, I'd like to remind you this is in the context of SMAD2 knockouts. So SMAD4 is hyperactivated. And then we ask the question, is SMAD4 a bad player or a good player under normal conditions? And I show you some preliminary data we have here that we delete SMAD4, use the same strategy in head and neck epithelium. And then this is one gene deletion that is sufficient to induce spontaneous head and neck cancer lesion. And then this is a histology just show you that adjacent skin epidermis is completely normal. And then the buccal epithelium is hyperplastic, then dysplastic, then transition to head and neck squamous cell carcinomas. So it's very similar to human head and neck cancer. And then they can also develop metastasis. And this is just validation of the metastatic lesion by keratin staining. So this is very surprising in a way because SMAD4 deletion or loss in colon cancer, pancreatic cancer is at the similar stage when TGF-beta receptor loss or SMAD2 loss. But from this data, it shows that SMAD4 loss can play both initiation role and promotion role in head and neck cancer. So this intrigued us. Go back, look at when SMAD4 is lost in head and neck cancer. So what we find that SMAD4 protein loss is actually occurring in adjacent mucosa of the head and neck cancer. So indicating it's a relatively early event. And also they can go, the mechanism is related to either hypermethylation or loss of heterozygosity. So still that there are not many examples that one gene deletion caused from all the way initiation to metastasis, and we suspect maybe these tumors have some genomic instability that cause lots of simultaneous genetic changes to collaborate. And then this is a CGH analysis, and this is an example of the genetic loss commonly found in human head and neck cancer regions. So a genetic level that this model mimic human head and neck cancer closely. And also in the stromal, again, we saw TGF-beta related paracrine effect. For example, Th17 cells, which are the pro-inflammatory T cells and requires TGF-beta for maturation and activation, is infiltrated in the SMAD4 knockout mucosa and head and neck cancer lesions. And angiogenesis also have increased ALK1, which is the type 1 receptor of TGF-beta in endothelial cells, indication of the TGF-beta related angiogenesis. So again, this is very similar to TGF-beta receptor loss, is a negative feedback loop. However, different from TGF-beta receptor loss, when the receptor is deleted, downstream signaling cascade get completely blocked, where SMAD4 deletion still have SMAD2 and 3. And these signals may still go through. So we look at the SMAD2 and 3 and find that SMAD2 is actually inactivated in these lesions, but SMAD3 has increased SMAD3 staining and also nuclear staining, so indicating that SMAD2 and 3 are differentially regulated. This is quite important because many people studying TGF-beta pathway often use phospho-SMAD2 as a surrogate marker for pathway activation, and this just shows that 2 and 3 may be quite different. So the question is that SMAD3 activation linked to TGF-beta-induced angiogenesis and inflammation? So we crossed the TGF-beta transgenic mice into the SMAD3 or SMAD2 heterozygous background. And this is from the skin lesion, very similar to head and neck lesion. In the parental transgenic mice, they have lots of infiltrated leukocytes and epidermal hyperplasia. Then in the SMAD2 heterozygous background, this is pretty much the same. But in the SMAD3 heterozygous background, you can see that leukocytes are gone, and then now epidermal thickness went back to normal. So indicating epidermal hyperplasia is mainly contributed by increased inflammation. So in fact, that the parental transgenic died in about 5 months of severe inflammation. The only way we can keep them life through normal lifespan is to cross to the SMAD3 heterozygous background. And how this related to cancer? In fact, that a few years ago, we studied SMAD3 knockout mice for carcinogenesis, and initially we thought they would be more susceptible to cancer, and actually they are more resistant to cancer, and they have a few experimentally induced papillomas, but none of them converted to malignant tumor. And the most striking phenotype is that they have reduced angiogenesis and inflammation. So in summary, what the data I showed you here, what we learned that similar to TGF-beta, individual SMADs can be a double-edged sword during cancer development. And SMAD3/4 dependent EMT promotes malignant progression. However, the EMT phenotype does not always associate it with more metastasis. For example, SMAD4 knockout tumors, they don't have EMT phenotype, yet they develop more metastasis than SMAD2 knockout tumors where they have EMT phenotype. And based on the several models I showed you here, and we think targeting tumor epithelium together with control of TGF-beta paracrine effect may provide a more effective therapeutic strategy for head and neck cancer. So finally, I'd like to thank my lab members. This was taken in Portland, Oregon before we moved to Denver. Shilong Lu was a postdoc that made the TGF-beta transgenic and TGF-beta receptor knockout mouse model, now moved to Denver as a tenure-track assistant professor, started his own lab. And Steve Markowski is a pulmonary fellow, worked on the SMAD4 knockout model, then now he's also a tenure-track assistant professor in Denver. Philip Owens started the SMAD4 knockout model study. Was a grad student, and right now moved to Hal Moses lab to be a postdoc there. And Sophia and Ruth are the MD PhD students, work on the SMAD4 knockout tumor model as well. And Christina is another MD PhD student, made the SMAD2 knockout tumor model. So I also like to thank the collaborators and then also people who helped us. Taylor sent us the KRAS floxed mice a very early stage, and how Moses provided us the TGF-beta receptor flux mice, and Chu Xiaodong provided us the SMAD3 knockouts and SMAD4 heterozygous SMAD4 flux mice, and Irvin Boltinger provided us SMAD2 flux mice. So I would like to thank you for your attention and stop here.